Welcome to Biochemistry 408. The title of this course is Blood Biochemistry and Immunochemistry. Blood Biochemistry and Immunochemistry. This is the 5th lecture series, and the topic is chemical basis for the functions of leukocytes and lymphocytes. Let's look at the introduction. Immune cells are produced in the bone marrow and circulated throughout the body via the lymphatic tissues. The human immune system is complex and are composed of two critical cell components, namely leukocytes and lymphocytes. Please take note of those two terms, leukocytes and lymphocytes. These are essential for maintaining health and defending the body against diseases. Understanding the biochemical basis for their roles could help us mitigate several diseases. Now, let's look at the objectives of this learning section. First, I told you a minute ago to take note of the word leukocyte and lymphocyte. At the end of this lecture, you should be able to define the two terms. Secondly, you should be able to describe the major role of leukocyte subtypes and explain the differences between innate and adaptive immunity. Fourthly, you can now summarize the biochemical mechanisms used by leukocytes. In addition, you will be able to discuss how monocytes bridge innate and adaptive immunity. Describe the function of B cells, T cells, and natural killer cells. Identify the biochemical mediators released by basophils and eosinophils. We will start by looking at some basic definitions. For example, we'll ask ourselves, what are leukocytes? Simply, leukocytes are white blood cells. They circulate in the blood and the lymph system. There are other subsets of white blood cells called lymphocytes. In the introduction few minutes ago, I told you to take note of leukocytes and lymphocytes. While leukocytes are white blood cells, lymphocytes are subsets of white blood cells. In general, they are all white blood cells. That is, both leukocytes and lymphocytes are white blood cells. Now, still on definitions of the term leukocytes. We have variants of the white blood cells, such as neutrophils, eosinophils, basophils. Neutrophils are the most abundant type of white blood cells that are crucial in fighting bacterial infections. Eosinophils are involved in fighting parasitic infections, and allergic reactions. Basophil also play a role in allergic reactions and inflammation. The other types of leukocytes, which in a sense are the lymphocytes. Recall, the topic of this present lecture is biochemical basis for the functions of the leukocytes and lymphocytes. And I just told you that leukocytes and lymphocytes are white blood cells. The difference is that lymphocytes are a unique subset of leukocytes. They include the monocytes and the lymphocytes. The monocytes are the largest type of white blood cells and differentiate into macrophages and dendritic cells, which are involved in phagocytosis and antigen presentation. Lymphocytes, on the other hand, are responsible for adaptive immunity and include the T cells, the B cells, and the natural killer cells. Now, these two cells, or these two white blood cells under leukocytes, can be classified on the basis of their granulation or agranulation. We have the granulocytes and agranulocytes. Granulocytes are those that have granules in their cytoplasm, while the agranulocytes are those that have invisible granules in their cytoplasm. The granulocytes include basophil, eosinophil, neutrophil, while agranulocytes include monocytes and lymphocytes. Of course, the lymphocytes, like I told you, are the B cells, T cells, and the natural killer cells. Let's move forward. Now, let's look at innate immunity. Since we are talking about cells that help to fight immunity. Now, this is the immune system we are born with. It is a body first line of defense. The key features are that they are non-specific. 2, they are immediate in action. 3, and they lack immunological memory. Now, let me expatiate further. The non-specificity involve the fact that this particular cell responds to all pathogens similarly. Immediate action implies that they are rapidly activated and finally, they do not have memory of the past or repeated exposure. We may like to know the components of innate immunity. These are all the manner of physical barriers we can think of, such as the skin, mucus, stomach acid, enzymes, as we have in tears, saliva block, or destroy pathogens. Of course, the cellular defenses, as we have in the phagocytes that engulf pathogens. with their cell membrane. The natural killer cells that destroys infected or cancerous cells. We also have soluble proteins, which form the complement system of proteins that also help in destroying pathogens. It is the turn of adaptive immunity. This is a type of immunity that develops over time. It offers specific defense and immunological memory. The key features of this type of adaptive immunity, or another word for it, is acquired immunity. They are specific, they are slower to activation, they retain or they are retained in memory by the body cells, so in case of repeated or future infection. Now, components of lymphocytes, there are the B cells, T cells, cytotoxic T cells, and the antibodies. The B cells produces antibodies that neutralizes pathogens or mark them for destruction. There's a subclass of T cells that are called the T helper cells or the helper T cells. The helper T cells help activate B cells and macrophages. B cells especially for the production of antibodies. So the B cells require T helper cells for antibody production. The cytotoxic T cells kill infected cells, while antibodies proteins bind to antigens, blocking pathogens or recruiting other immune system. Now, this is a table shown that contains the type of leukocytes, including lymphocytes, their main role, and their distinct functions. For example, we can say that neutrophil is the first responder to infection caused by bacteria, especially. The distinct function involve phagocytosis, and generation of reactive oxygen species to kill pathogens. Lymphocytes is an adaptive immunity or specific immune response. Distinct function: B cells produces antibody to neutralize pathogens. T cells destroy infected or abnormal cells coordinated by immune responses. Natural killer cells kill virus-infected and cancerous cells and provide immunological memory for faster future responses. These ROS or reactive oxygen species, along with other neutrophil-derived antimicrobials, damage and kill the ingested pathogens within the phagosomes. Eosinophils, on the other hand, bind to parasites through antibodies such as immunoglobulin E or the complement proteins, marking them for destruction. Upon binding, it releases its granules, which contains toxic proteins such as the major basic protein MBP, and the eosinophil cationic protein that damage the parasite cell membrane and disrupt its cellular processes. It is also involved in complement activation, which can also lead to generation of anaphylatoxins, which can further enhance the inflammatory responses and eosinophil mediated killing. Basophils. These cells contains heparin. Heparin is a substance that prevents blood from clotting too quickly. This is especially important at injury sites where a blood clot could interfere with the flow of blood and the delivery of nutrients needed for healing. Still on basophil, it releases histamine that causes the blood vessels to dilate, increasing blood flow to the area. This increased blood flow delivers more oxygen and nutrients to injured tissues, helping to promote healing. Still on basophil, the combination of histamine and heparin helps to create a controlled inflammatory response at the injury site. This inflammation is a normal part of the healing process, as it helps to clean debris, fight infection, and stimulate tissue repairs. Monocytes. Upon encountering a pathogen, monocytes engulf and digest it through a process called phagocytosis. This process involves the cell membrane extending around the pathogens, forming a vesicle called phagosome. Within the phagosome, enzymes digest the pathogen, breaking them down into smaller antigens or pieces. Monocytes can also differentiate into dendritic cells or macrophages, both of which are types of antigen-presenting cells. Take note, APC is antigen-presenting cells. These antigen-presenting cells take the processed antigen and display them on the surface or on the cell surface, specifically on molecules called major histocompatibility complex. The monocytes are also involved in activation of T cells by forming antigen major histocompatibility complex that is recognized by T cells. This complex are crucial part of the adaptive immune system. This recognition initiates a cascade of events that activate the T cells, leading to the production of cytokines and other molecules that help eliminate pathogens. Let's consider the link between innate and adaptive immune system, for which monocytes play a key role. By acting as antigen-presenting cells and triggering T cell activation, monocytes effectively bridge the gap between the innate and adaptive immune system. This ensures a robust and targeted immune response against pathogens. B cells are the primary players in the humoral immune system, producing antibodies that bind to antigens, neutralizing them or marking them for destruction by other immune cells. They also differentiate into memory cells for long-term immunity. That is, after synthesizing antibodies, now they are stored in case of future exposure. They can easily reproduce the antibodies. T cells. These are cell-mediated immunity. Cell-mediated immunity. It is crucial against intracellular pathogens and cancer cells. They are broadly divided or categorized into T helper cells or cytotoxic T cells. Now, the T helper cells are the CD4 plus cells, while the cytotoxic T cells are otherwise referred to as CD8 cells. So when you hear CD4, we are talking of T helper cells, while CD8 are cytotoxic cells. Now, the CD4 cells orchestrate the immune response by producing cytokines that activate other immune cells, including B cells and cytotoxic T cells. On the other hand, CD8 or cytotoxic T cell directly kill infected or cancerous cells by destroying them and preventing their spread. So, question for you, what are cytokines? What are cytokines? Let's look at the natural killer cells. They are part of the innate immune system or natural immune system and are characterized by their rapid response to infected or cancerous cells. They do not require prior exposure to a specific antigen like B and T cells. They can still target and kill a wide variety of cells, preventing the spread of infection and potential cancer cells. Now, in conclusion, we have seen that leukocytes and lymphocytes are central to immune defense, each subtype executing specialized biochemical functions. Their coordinated actions ensure both immediate and long-term protection against a wide range of pathogenic organisms. I have here some study questions for you, which you can lay your hands on to test your own understanding. Thank you. And that brings me to the end of my own section of BCH 408. Now, In case there is any of you that do not understand one aspect or the other in the areas I have taken, you can help yourself by noting your questions and sending it to the class rep, who will in turn forward them to me, and I will address them through a video to you. Meanwhile, prepare yourself for a test soonest. I will get in touch with you as to when the test is coming up, and the type of test, whether it's going to be OBJ, short answers question, or paragraph. That it will be made known to you. Meanwhile, have a nice day. Thank you.